Before we can start this procedure of PCR, we have to do some homework. In order to amplify any DNA segment using a set of primers, we have to find out the temperatures. We have to work to find out the number of cycles required. This is referred as programming of PCR. PCR consists of three elements. Denaturation, that is the separation of double-stranded DNA. The two strands of DNA, they get separate. After the separation or denaturation, the second step is attachment of the primers, which is called as annealing, to the single-stranded DNA. The attachment of primers is crucial for the third step, which is extension. Once the required primers they attach to the template strand, then the extension is possible and amplification can be seen. Denaturation is achieved in PCR through temperature. What is the length of the DNA? What is the base composition of the DNA under investigation matters a lot. The temperature, the denaturation temperature, is determined by GC contents of the DNA, that is guanosine and cytosine. Higher is the GC content, higher will be the denaturation temperature. This is because of the reason that G and C, they are base pairing through 3 hydrogen bonds. Longer is the DNA, greater time will be required for its denaturation. While performing this method, if the temperature is not calculated correctly and it is low as we should have used, it will cause 80 rich regions to separate, but the denaturation of full length of DNA will not be achieved. And if the temperature is low, re-annealing can also happen so that DNA goes back to its native form. While working with the Taq polymerase, the denaturation temperature which is practiced or which is used is 94 to 95 degrees centigrade. Taq polymerase is the only enzyme which can withstand such an high temperature for 30 cycles or more cycles of PCR. For a standard PCR reaction, the denaturation conditions with the DNA sample having GC contents 55% or less, time for denaturation should be 45 seconds at 94 to 95 degrees centigrade. If the GC contents are more, then there would be problem or we need more time for denaturation. After the denaturation, we have to talk about annealing temperature. As from 94 degrees centigrade, we have to drop the temperature, and the annealing temperature is critical because that determines if the primers bind to the template strand or not. Too high annealing temperature, poor annealing of the primers. Too low annealing temperature, non-specific annealing, means the primer will bind to non-specific sequences and will try to amplify those areas which are not our interested areas. Annealing temperature is calculated keeping in view the melting temperature. The melting temperature is the temperature at which the primers separate from the DNA template. Annealing temperature should be 3 to 5 degrees centigrade lower than the melting temperature, and if still the results are not clear, we have to try a range of temperature 2 to 10 degrees centigrade below the melting temperature. We have to optimize the annealing temperature in multiple PCR reactions, keeping in view the sequences of bases in the primers. Then the third step is extension, which is achieved at 72 to 78 degrees centigrade because this is the best temperature range for working of Taq polymerase. Once we program the PCR and we set it up and we start it, then we have to see from which cycle of the PCR we will get our required DNA or DNA fragment amplified. This figure shows you the first cycle, the template DNA. Then we have the red color area, that is our gene of interest. This portion is going to be amplified because we have designed our oligonucleotide primers keeping in mind the boundaries of this gene. In the first reaction or in the first cycle, we can see that the two parental strands, they are denatured, primers annealed, extension of these primer occurred. But towards the end of first cycle, the DNA will keep on extending because we already have the template available. By the time the second cycle is going to start, by increase in temperature, these two daughter molecule or newly synthesized DNA molecules, they are going to denature again. Now look at this picture. This is the first molecule, this one is the second molecule. Again, denaturation is going to happen. This is the parental strand here, this is the parental strand here, and the new strands are synthesized. In this picture, you can see still one of the strands is longer than the originally required DNA fragment. This is the third cycle of PCR when we are going to get double-stranded DNA of our required gene or the DNA fragment exact length. And from third cycle onward, there will be increase in number of that specific DNA molecule. Taq polymerase works better and the best at 72 to 78 degrees centigrade. This is the temperature range where we can optimize it to get the good amplification. The polymerization rate of nucleotides is 2000 nucleotides per minute under these conditions at this temperature. As a thumb rule, we should carry out the extension for 1 minute for every 1000 base pair product. In some labs, the last cycle is extended for extension, and the idea is to maximize the extension of the DNA strand from reading the template strand. But overall, if it has been reported, overall it has been seen that it is not going to make any significant change in the results. How many number of cycle should we plan at the beginning of the PCR? It depends on the quantity or number of DNA molecules in the sample. That helps you to decide whether you should go for 30 cycles, 20 cycles, 25 cycles or more. As a general rule, with a single copy DNA, minimum 25 cycles of polymerase chain reactions are required to get some noticed or to get some acceptable level of amplification.